Today's topic is strategies for iron uptake in plants. Iron is one of the nutrients that are indispensable for plant growth and development. It also takes part in many chemical reactions, including photosynthesis and respiration, as it is redox active, owing to its ability to easily accept and donate electrons. Iron deficiency usually leads to a reduction in plant viability. Despite its importance, iron can also be toxic when it accumulates to inappropriately high levels. This is because free iron can participate in the Fenton reaction and generate cytotoxic hydroxyl radicals. Fenton reaction? Fenton reaction describes formation of hydroxide and hydroxyl radicals by reaction between iron and hydrogen peroxide, right? Plants have evolved sophisticated mechanisms for tightly controlling the influx of iron to maintain iron homeostasis. Next is iron uptake. Plants use two different strategies for iron uptake to adapt to iron deficient environments, strategy 1 and strategy 2. Strategy 1 is a reduction-based mechanism and strategy 2 is a chelation-based mechanism. Reduction-based strategy 1 involves acidification of the rhizosphere. Rhizosphere, if you remember, I have already told you in our previous lecture, rhizosphere is the region of the soil in the vicinity of plant roots, in which the chemistry and microbiology is influenced by their growth, respiration, and nutrient exchange, right? So, this strategy 1 involves first acidification of the rhizosphere by root-released protons. Then reduction of ferricoins to ferrosoin by membrane-localized reductase and root uptake of ferrosoins by membrane-localized high-affinity iron transporters, right? Notably, ferricoins bioavailability decreases 1000-fold with an increase of 1 pH unit between 4 and 9. The low efficiency of FRO2 protein for reduction of ferricoins in alkaline soil results in the failure of strategy 1. Strategy 2 then is a chelation-based mechanism. It is well known in gramineous plants, right? First, we'll discuss reduction-based strategy. In response to iron deficiency, protons are released into the rhizosphere by AHA proton ATPase expressed in the epidermis. This lowers the soil pH, making iron more soluble. When protons are released into the rhizosphere, this lowers the soil pH, making iron more soluble. Then just look at this diagram. AH2, AHA1, AHA2 and AHA7 are all upregulated in the root epidermis in response to iron deficiency. Now, what is this AHA? A, Arabidopsis, H, protons, and A, ATPases, right? Actually, these form a family of proteins in plants. For example, in Arabidopsis, there are 11 functional plasma membrane proton ATPase family members, denoted by AHA1 to AHA11, right? So, AHA2 is the primary root proton ATPase in the iron deficiency response, right? So, it releases protons in the rhizosphere, right? expression level of AHA2 is the highest among the three, means AHA1, AHA2, AHA7, right? And only the loss of AHA2 was found to reduce rhizosphere acidification during iron deficiency. Then the NADPH dependent ferric chelate reductase, ATFRO2, this. Here ATP Arabidopsis thaliana, right? So FRO2. This is NADPH dependent ferric chelate reductase. It then reduces this ferricoins to ferrosoins, right? And electrons then are transferred from NADH plus across 4 heme groups to iron in the rhizosphere. Once reduced, these ferrosoins can then be transported into the root epidermal cells by the divalent metal transporter IRT1, right? IRT1 is a divalent metal transporter present in the epidermis. Actually, another aspect of iron deficiency response in non-gramineous plants is the secretion of phenolic compounds into the rhizosphere and the uptake of apoplastic iron, right? It has been observed that as much as 75% of the iron in the roots is attached to the apoplast, as the negatively charged carboxylic group in the cell wall serve as the cation sink. So this pool decreases when the plant becomes iron deficient and suggesting mobilization into the symplast. So phenolic exuded by the root in response to iron deficiency, and it facilitates the utilization of apoplastic iron and and the recovery from the iron deficiency, right? Now, next is chelation-based strategy. This chelation-based strategy, as I already told you, it is very common in gramineous plants. So grasses depend on the uptake of iron chelated by soluble cytophores with a high affinity for ferricoins, right? Now, just look at this diagram. In response to iron deficiency, musonite acid family phytosidrophores, PS, musonic acid family, MA, phytosidrophores are synthesized from L-methionine and released from the root epidermis into the rhizosphere, right? This PS radially chelate ferricoins and the phytosidrophores and iron complex is formed. This complex is then, this is complex. This complex is then transported into the root by member of YS or YSL family. YS means yellow stripe. YSL yellow stripe like genes. So here YS1 in maize and barley and OSYSL15 in rice. So actually the chelation strategy is less sensitive to pH than reduction strategy. And there is a strong correlation between the volume of PS, means these phytosidrophores released and resistant to iron limiting soils. For instance, barley, which is adapted to alkaline soils, release a much greater volume of PS than most of the rice species, which are adapted for growing in anaerobic soil, where iron is more soluble, right? So within the epidermis, divalent metal efflux means FPM2 is expressed during iron deficiency on the vacuolar membrane and may serve into buffer iron uptake by sequestering excess free iron in the vacuole, and the iron may be bound by phyllate in the vacuole. Then iron move out of the epidermis via the plasmodesmata, right? Third is combination of reduction and chelation strategy. Gramineous species, rice, for example, combines components of reduction strategy uh, seen in non-gramineous plants with iron PS uptake. Of the 18 yellow stripe-like genes in rice, OSYSL15 is the primary transporter responsible for uptake of iron PS from the rhizosphere. OSYSL15 is upregulated in response to iron deficiency and is expressed in the plasma membrane in the root epidermis, in addition to the steel flowers and developing seeds. Rice produces much less PS than maize and barley, making it less tolerant to calcareous soils. Rice compensates by expressing the divalent metal transporters OSIRT1 and OSIRT2 in the root epidermis in response to iron deficiency, right? In fact, in rice mutants, without the ability to synthesize PS, these iron transporters were found to be dramatically upregulated in the roots, 30-fold for OSITR1 and 64-fold for OSIRT2. This compensated in terms of iron uptake in waterlogged soil where ferrous iron is readily available, right? In aerobic soils where iron is limited and the hydroponic solution where only ferrous ions was supplied, mutant plants died. It has been proved experimentally, right? Thus, the reduction strategy alone is inadequate in ferrous limiting condition. Right? Thanks for watching. If you like this video, make sure to subscribe for more. Thank you.